in the future as well. So for my project, I needed expertise in photogrammetry, which is one of the methods that we used, and that was available at Tritonia. So fortunately, both Tritonia and SAMS were partners in Assemble, and they're located on the same premises on the west coast of Scotland. I needed some sort of dry workspace for constructing and programming the instrumentation, some laboratory facilities for sample processing. Naturally, I also wanted to access an interesting site for this project, which was, we decided to study a site in Lough Crearan, which is a marine protected area on the west coast of Scotland. We needed a small research vessel and naturally also diving facilities and a dive team to help me with my work, but also to perform the photogrammetry work. And all of these were available at SAMS and at Tritonia. So the photogrammetry method is a recent and evolving technique that allows us to, through imaging, reconstruct habitats. So create sort of 3D scale 3D models of habitats in great detail. And this is done following by taking images of the seafloor in an organized manner. And you can see there a picture of a diver doing a survey along a marker line on a reef. And the camera configurations can range from small cameras such as GoPros to larger DSLRs with external lighting. It depends on really on the habitat you're trying to image and the resolution that you're trying to resolve. But it takes around 1,000 images to reconstruct an area of about 100 square meters. And you can see an example of one of the what's called a dense point cloud on the bottom right. And the blue squares show the position of the individual images. And anyone who's interested in this method should look at this recent paper by Bailey and Mogg, which outlines in detail the protocol and the requirements for this method. So once you have the dense point cloud, you can generate, you can do different things with it, but people have been more and more using this for biodiversity studies. In particular, things like species identification using manual or machine learning techniques. You can look at things like habitat coverage. You can look at the size, the abundance, biovolume, and estimate the biomass. And you can also calculate something called the rugosity, which is a measure of terrain complexity. And the rugosity is important ecologically because it gives you an indication of the habitat structure, and the structure of the habitat creates sort of increases the surface area over which biological processes and interactions can take place. And it also affects the hydrodynamics within the benthic boundary layer. So the other approach that I wanted to use was aquatic eddy covariance, which was my expertise. So in a way, I was bringing my expertise to SAMS and to Tritonia, and I was taking advantage of the expertise that they had over there. And this is another sort of recent and evolving method, and it's a way of quantifying benthic fluxes and productivity on a habitat scale, so on tens of square meters. So the idea here was really to link to that we have these two methods that can each do what they do on the scale of whole habitats. And we can then merge these two methods to try and to inform us about the role of biodiversity within the productivity of benthic ecosystems. And this method is also completely non-invasive. It does not interfere with organisms, sunlight, or the flow velocity. And the hardware consists of an acoustic velocimeter. You can sort of see the images of the complete system up top on the top right. And one or more fast response oxygen microsensors. There's those sort of needle type sensors on the top right. And the idea is that you record the vertical velocity of the water and the oxygen concentration at high frequency to capture all of the flux contributing turbulent eddies. So the flux of oxygen between the seabed and the water is mostly driven by turbulent exchange. And this system allows you to capture all of those turbulent eddies and calculate a flux. The instrument also records additional parameters such as water depth, flow velocity, direction, and signal intensity. And if you look at the image on the bottom right, you can see sort of a cartoon of an instrument on the seafloor. You can see that the flow direction is coming from the right and going to the left. And that sort of red area that you see on the seafloor is the area that's being, that is contributing to the flux that is measured by eddy covariance. And this area is located upstream of the instrument. So if you have a tidal flow, for instance, you're going to interrogate two different areas of the seafloor in your measurement. We can also see that the flux contribution within this area is not linear. So most of this, while the instrument integrates an area typically of between 30 and 100 square meters, most of the flux originates from an area around the instrument of, say, the nearest 5 meters. So this makes it possible for us to look at the habitat and the biodiversity around the instrument to try to get a handle of what biodiversity there is that is contributing to this, to the productivity signal that we measure with eddy covariance. So the first step was to bring the equipment to from Denmark to Scotland. And once we're there, we waited for a good window of weather to do some exploratory dives and to locate our study sites within Loch Ruaran, within a sea loch. And the site that we wanted was, it's a protected site composed of the boreal mussels, modulus, the horse mussels, which are quite large mussels and form these reefs that are incredibly biodiverse. Once we had located a site that was several hundred square meters in size, we marked it and we deployed the eddy covariance instruments by slowly sort of lowering it onto the seabed. And then my colleagues at Tritonia, Andy Mogg and his colleagues, proceeded to carry out the photogrammetry survey to image the whole area around the instrument. And we revisited the sites 3 times in a week just to make sure that the instrument was recording as it should and to collect some additional data as well. You can see in the image on the top right that the water was actually quite turbid. So the photogrammetry method is quite useful in turbid environments as well. So the eddy covariance deployment produced 54 hours of data. So we can see in the two windrows plots on the right, the center point is where the instrument is located. And then I binned the fluxes depending on the direction that they were coming from. You can see that the fluxes originate from different directions of the reef because of the tidally driven flow. And around half of the fluxes originated from the east side of the lander, and the other half originated from the west side. All of the fluxes were negative, suggesting that there was an uptake of oxygen by the seabed. And there was very low sunlight intensity reaching the seafloor, less than 10 micromole photons. And we could not see any light response in the fluxes, meaning that if there was any photosynthesis taking place, it was not sufficient to influence the magnitude of the fluxes that we measured. So this kind of facilitates the interpretation of our data because it allows us to interpret the fluxes entirely within a context of respiration. So we can look at the respiration of the reef. The photogrammetry model converged successfully. So it took just over 1,000 images to create this model. And Andy was also successful in generating some additional information from this point cloud and isolating the reef mounds and look at the distribution of where these mussels were. So if you look in the top image, this is actually the point cloud looking down on the instrument from above, and we can see a sort of mosaic of dark and light patches. and the dark patches are the modulus reef itself. And by using certain routines, Andy could sort of isolate this portion, so the reef coverage from the rest of the sand and gravel that sort of interspersed the reef. So we could process this. Andy could process the data in a similar way as I processed the fluxes, and so we could quantify the reef volume in different directions. We could quantify the geometric rugosity, the rugosity of the reef. We could also I'd calculate the mussel biomass in different directions by getting the length of the mussels and then using literature values to convert that to biomass. Naturally, we didn't want to extract the mussels from the reef to do that. We just wanted, we didn't want to disturb them at all. And we could also calculate the reef area, so the reef coverage, so to say, which typically range from 50 to 60%. And all of this, all of these models are georeferenced. We know where the location is. We could revisit these sites, and I think this is a real strength of the photogrammetry method. What I was particularly excited about was that we could try to link our metrics of biodiversity, our measurements of biodiversity, with the data from the Eddy Covariance instrument, the productivity of the ecosystem. And here we see a plot showing that sort of the increasing number of mussels drove a higher oxygen flux and also resulted in a higher rugosity. So the mussels are creating a habitat, right, and for all the microbes and other organisms to colonize. And this is driving a higher benthic oxygen uptake rate. And all this was done from a single instrument deployment. So despite it being a relatively small data set, I think it really illustrates the strength of this approach. There are some limitations with the photogrammetry approach. Naturally, if there is something that is moving, then this is going to reduce the resolution in that particular part of the model. But both photogrammetry and Eddy Covariance are evolving techniques. So we can focus some of our efforts in the future to try and tackle some of these limitations specifically. I just want to finish with some overall thoughts about my experience with Assemble Plus. So the first point is that Assemble Plus really made the project possible. So I wouldn't have been able to go to Scotland had it not been for Assemble Plus. And I really felt quite spoiled in a way. I was given lots of access to really nice facilities and expertise that I could sort of take advantage of. And the local points of contact at both SAMS and Tritonia really facilitated the process and made it seamless. There was I felt like I had a generous accommodation and food budget, which really makes the experience more pleasant. And I took the opportunity to present a seminar at SAMS during my stay, which turned out to be a great networking opportunity and has resulted in some further collaborations. My only gripe, perhaps, is that the project only covered shipment costs one way. So it sort of reduces the scope slightly in that this will be sort of a totally independent project, and especially thinking about younger scientists, PhD students, young postdocs, this could be a limitation for them. But overall, it was a great experience. And thank you for listening in.
Thank you, Carl. That was very interesting. And in fact, also, I'm glad because, you know, we had, well, we have what we call a GRA. Basically, it's to try to develop further some of those kind of technologies. And of course, SAMS and Tritonia are the leading partners in that process. But it's nice to see that, you know, it's being used in the project. What I've got a curiosity, well, maybe it's not just curiosity. So you showed what you did, and you had that rectangle. I mean, what was the area of that rectangle? Because clearly one of the limitations, seemed to me, was the small area that it was possible to cover with over 1,000 photographs. So how much more could you do?
Yes, so I think that area was just over 100 square meters, 130 square meters or something like that, which we thought would be sufficient, at least for extracting the information that we needed. I think one important point would be that you can achieve this within, say, one or two or a few dives. You know, you don't want it to be something that you're going to spend an entire week working intensively on, because you have a lot of other things to do. But so I think one of the strengths of the method is that in maybe one or two dives, you can actually cover the area that you need to investigate. But naturally, if you have more time to do it, then you could, you know, look further afield. Yeah.
And how much computing power do you need to do that? I mean, is it something that is accessible, or?
It is, as I understand it, and it does require significant computing power. I think our colleagues at Tritonia have some pretty powerful desktop computers. I'm not sure exactly about the details. I think Andy Mogg outlines all of the details in this paper that I quoted, and gives some details about the software as well that they use to do that. But it's becoming, I think, more and more accessible now. Yeah.
I'm just reminding the audience that you can ask questions. If you can, you know, just say something in the chat, and I'll ask you to talk. I still also have a comment, because you made, you know, I'm glad you make some of those comments about the Ensemble, in particular the last point, because, you know, our perspective has always been, you know, some co-responsibility, let's say, between the, you know, the two sides. And so that's why, you know, the money doesn't stretch all the way as much as you like.
Yeah.
It's more like a leverage. But I think you made, you know, some point. It may be some cases where, you know, this may not work so well, because, you know, either because people don't have that amount of money that they need, or because they are young scientists and so on. Of course, we assume also that PhD students have a supervisor that has the money, but it may not be the case. But that's it.
Yeah, that was perhaps a little cheeky comment, but
no, it's fine. I mean, it's fine. It's good to know. We are always ready for the feedback. In fact, everyone has to write some questionnaire at the end to give feedback on the visits and so on. Yeah. So we still have. Oh, there you go. Christine, you can chat. You can say it aloud. Christine Campbell.
Okay. Yeah. I was just saying that of all the
congratulations, by the way.
Oh.
The news reached Portugal.
Oh dear. Okay. Thank you. Yeah, I was just going to say, most people who came to SAMS didn't have the huge amount of kit that Carl had to transport, so he was exceptional. And
yeah,
the transporting thing, shipping limitations weren't a problem. But I'm glad it worked out well.
Yeah, and thank you for helping with that as well. I think you really made my life much easier.
Good.
Okay, there's another question by Forster Stefan. I'm not sure which surname is, but there you go.
Hi.
Hi, Stefan.
I just jumped into this meeting without being a member, really, but my first talk that I attend. Thank you, Carl. Very interesting. Getting right to that 3D plot, second last or last overhead you showed. Now, it's kind of clear that with increasing biomass, the total oxygen uptake should increase. But it isn't clear because it's 3D. I don't see how much does rugosity add to that. Do you have some more specific information on that?
Yes, I haven't looked at the individual components, the effect of it's something that I finished quite recently, looking at, unfortunately. Sorry, so I can't offer more information there. Yes, and I don't really know what to expect, honestly, as well. Like I don't really know, but I guess what you're suggesting is that I should decompose those sort of relationships and try to figure out how much one is contributing relative to the other.
Well, I would think that then this combination that you showed us of instruments is or approaches, that's one of the things that you can really learn from it, because we all want to know, more biomass, more oxygen consumption, but what if the biomass is the same, but the 3D structure is more complicated? How does that affect it? So that's actually going to, or it looks as if there will be some nice information in the future. Thank you.
Thank you. And I agree. I think it is a powerful approach in that sense, and we should just try to use it more. Do it in different habitats and see what sort of relationships we find. We don't really know what to expect, I guess, but or we have some idea what to expect, but maybe not what that relationship will look like specifically.
Mm-hmm. Thank you. I'm sorry. Last night I sent a message, but it had the wrong timings. In fact, Carl was the one that made me aware of it. So I hope that everyone managed to come and listen and see these presentations. Just as information, I can tell you that normally we get about 100 people, listening and watching. So that's not too bad at all. Although the total number registered for the conference is much higher, but it's pretty good, I would say. So the next will be in 5 minutes. So now we have a short interval since there are no more questions. And I would like to thank Carl for having positively responded to our request to give a talk, and for such a nice and interesting development. Thank you.
Thank you.
So in 5 minutes, we'll be back.
At NHBS, our purpose is to support those who are passionate about wildlife, ecology, and conservation. We stock a variety of books and equipment to suit the needs of marine conservation professionals, and our innovation and research team are here to help develop custom products for any project. We are happy to provide advice and to support you before, during, and after your purchase. Visit nhbs.com today to find out more.
So welcome back. So we're going to the next talk. The next talk will be by Imke Lang. She's at the University of Applied Sciences in Bremerhaven. And she also visited SAMS. So this afternoon we have a SAMS mainly talks. Identification of optimal cryopreservation conditions for the red microalgae, Porphyrium androdella. So please, Imke, can you start?
Yeah, can you hear me?
I can.
Okay. So I think, are you able to see my presentation?
Yeah, you just have to click. That's it.
Is it visible like?
It's just normal.
Okay, great. Okay, so I'm gonna start. Yeah, thanks for having me here today, and thanks for the organization of this nice conference. Very diverse presentations and topics. And I'm gonna speak about cell banking of red microalgae. Do you still see my video here? Can I remove it like this? I think now it's better for me at least. Okay, so cell banking of red microalgae. And yeah, as I just said, we've been in Scotland last year at the CCAP, Inzar, Manot, and myself. And we established, or we wanted to establish, a cryopreservation protocol for the red microalgae. And after returning to Bremerhaven, we had the lockdown and nothing could be continued. But since last autumn, we have a master student, Tintin Chu, who is continuing our work. And I like to present at least some preliminary results and thoughts on the cryopreservation. Yeah, so what are the questions for today? First of all, I like to ask the question what the concept of cell banking is and how we can overcome cryo injuries. And you might be curious why we in Bremerhaven are interested in red microalgae and what road to a standardized cryo procedure we are taking. And as I said, it's preliminary results. We are still working on that topic. So I will also include some steps that we are going to complete next. Yeah, so what's the concept of cell banking? In general, cell banking allows a high quality and standardized commercial production process. So if you have a production strain selected that might produce a very high amount of lipids, you like to preserve that characteristic. And you can do that by cryopreservation. And the concept of cell banking in a commercial context is that you have a master stock and a working stock. I'm gonna quickly activate the laser pointer. Okay. So you usually start with your selected cells or clones, and you cultivate them to a certain density, and then you harvest. And the harvest cells are cryopreserved in a master stock. And from that master stock, you thaw just one vial and start all over again, subculturing to, again, a certain density, and then you cryopreserve again. The difference here is that you have a higher volume and way much more vials, can be more than 100. And that working stock is then used for the scale up in your commercial production process. So a master and working cell bank actually is meant to prevent genetic variations of your target strain. And it also should avoid contamination. So you always have high quality, at least during the scale-up process. They have all the same age and also belong to the same genetic population. And by developing these protocols, we should consider to ensure a high viability after thawing, because stress and low viability after thawing also indicates that the population might be very diverse and we have higher probability of mutation, and that's something we want to avoid. And quick growth for scale-up is also important. So we should keep the cells in a physiological state that enables them to regrow quickly. So reduce lag phase after thawing and obtain biomass within a short period of time. So by using cryopreservation, we of course also have some critical steps, and that's the cryo injury that can be caused by ice crystal formation and also oxidative stress. And all these steps and also strategies to avoid cryo injuries are depicted in this graph here. So we have our happily growing culture, and by cooling it down to sub-zero temperatures, the extracellular osmolarity is increasing and the cells are dehydrating, the more ice crystals are formed. And then it depends how quickly you cool down your sample, either slow or rapid or optimal. You can increase ice crystal formation and basically dehydrate the cell, and then it's not viable after thawing. You can also get intracellular ice crystal formation, and then you have mechanical stress and injuries that also reduces viability after thawing. So that can be inhibited by cryoprotecting agents, CPA. Or these can either control the dehydration of the cell during the freezing process, or they increase or basically promote vitrification of water, which also reduces the injury and dehydration by ice crystal formation. Optimal cooling is some kind of an intermediate. The cells lose a bit of water, but they are still viable. And they can then store it at ultra-low temperatures, either in the liquid or gas phase of liquid nitrogen. And while we expect to have no thermodynamic reactions and indefinite storage. Thawing is another risk during cryopreservation. Recrystallization and devitrification can injure the cells and reduce viability. So optimal warming rate is important. And then the last thing to think of and to consider is the toxicity of the cryoprotecting agent. Rapid removal of CPA is recommended by literature, and that's something we need to address when we develop a protocol for a certain alga strain. So these are the main things we have to consider: controlled rate freezing and thawing, and the CPAs and other additives that might be necessary. So why are we interested in red microalgae? Well, we have a project currently running in Bremerhaven that deals with algae-based lubricant additives for tribological processes. That project is called Albina, and it's a big consortium of different partners from universities and institutions. And lubricant additives are very versatile and have different applications. And the ones that we find in red microalgae, these molecules might replace additives that act as antioxidants and antimicrobials, or directly, like the sulfated polysaccharides, act in the tribological process, so the interaction between two surfaces, in this case in the metalworking process. So that's why we are looking at red microalgae, and we like to preserve them, to keep them in liquid nitrogen, or at least at minus 80 degrees, to have our cells healthy and preserved. So the aim of the project was, and still is, the development of a simple and reliable cryopreservation procedure for red microalgae strains, resulting in a high post-thaw viability. So very ambitious. And we started by looking into literature. Well, there's a lot of protocols available, depending which algae are in focus. A lot of protocols are available for green algae, and there are also some available for red microalgae. Most of the culture collections don't have a cryostock, at least at CCAP. At that time, there wasn't. So and as you see, these are the main hits that you get if you enter certain keywords in the search engine. So one publication from 2006, cryopreserved Porphyridium cruentum, successfully in 5% DMSO using a two-step cooling protocol. Well, they reported a very high viability, and interestingly, in an uncontrolled freezing process. The viability was determined by oxygen measurements. And the next publication we found was 3 years later. And they also successfully cryopreserved Porphyridium aerugineum. That strain was cryopreserved with encapsulation, and they reported a post-thaw viability of 64%. Well, and then there are protocols available from the ATCC cryopreservation or the strain collection. And these protocols are standardized. At least they give very detailed information on that, but they don't give any details on post-thaw viability. So we don't know actually how high the percentage of strains after thawing is. So that was kind of our start. And Based on the literature, we had our own road to the standardized preservation procedure. And we first of all wanted to take a look at the pre-cultivation conditions, including the growth phase, when to harvest for the cell bank. During cryopreservation, well, the cooling wasn't something we wanted to look at, but the cryopreservation agents, CPAs, and also the cell density within the cryovial. And for the post-thaw treatment, we decided to figure out if centrifugation for removal of the CPA is an option, or if we rather dilute the sample after thawing. Because we have limited infrastructure in Bremerhaven, we don't have a controlled rate freezer machine, so we just have the Mr. Frosty. And we also don't have much other, let's say, instruments that help us to measure different parameters. So we decided two-step standard cooling protocol is simple, easy. Mr. Frosty is easy as well. For post-thaw growth, we are using 6-well plates to test a lot of different conditions. And we keep the samples after thawing in the dark for 12 to 24 hours. And then they are subjected to light, 50 micromole photons per square meter in second. The viability is tested in 2 ways. First of all, we detect the viability with a cytox stain in the flow cytometry. That is done 24 hours after thawing. So we give the cells a rest before we analyze them. And cell growth is the other measure, basically, for our viability. So here are 3 examples of the in total 12 strains we received from the CCAP. We had all 12 strains in our investigations in Scotland. They were all xenic, so bacteria, and I think in one there was also fungi were present. So that makes cryopreservation a bit more complicated. And we started to purify the strains, and we are still working on that, but the majority of the strains is xenic by now. So in Scotland, we started with 5 strains that we selected within the first days, because these were the strains that grew best and gave us the opportunity to finish the experience within that 4 weeks we stayed there. So these 5 strains represented Porphyridium and Rhodella, and we grew them in small bottles of 150 milliliter of medium, and the conditions are here. We had an addition of 2 millimolar bicarbonate 5 times a week as the carbon source. So on the right-hand side, you see the growth curves. And you see that Porphyridium strains grew best compared to the Rhodella strains down here. So they are not really quick grower, at least not under these conditions. And the best was Porphyridium sordidum, CCAP 1386. So the arrows indicate the sampling days. And on these sampling days, we either tested all of the conditions, and I think at the latest point, INSA also tested the encapsulation method. And this is again shown here. So we tested several parameters and wanted to know which one impacts the success of cryopreservation. So sampling at different growth phases, and then different concentrations, 5 and 7.5% DMSO or methanol, and then the encapsulation method. We also differed the density in the cryovial, and that was between 0.5 and 4. And after thawing, we always did a 1:10 dilution for the revival. Well, and what can I say? Unfortunately, we weren't lucky. Only 1 strain could be revived, so grew after all our treatments, and that was Porphyridium purpureum 1385. So 2 sampling days apparently were perfect for that strain, and we had several conditions. Well, then we left, so we couldn't continue with that. And back in Bremerhaven, we had the lockdown, several months went on, and then we decided to continue. So currently in that master thesis, we have studies included that address the tolerance for the cryoprotectants. So we will test, or we are testing more than just dimethol and methanol. We establish a flow cytometry-based day-at-life assay. And we continue these experiments with other strains and only focus on rather low densities in the cryovial instead of having that big range. And now we are also able to compare axenic and xenic cultures. A lot to do. Yes, so I'm going to start with live dead analysis using cytox stain and flow cytometry. That is a quite easy assay. Cytox is a stain that is available in different fluorescent colors, and we have the green color here. And this stain passes permeable membranes. So you can detect cells that are dead or injured, and all living cells won't fluoresce. So that's basically the principle of this assay. So we have 3 plots here. And what we did, we harvested a culture in a late exponential growth phase and treated the cells either directly with the stain, or we killed the cells by cooking them for quite a while at 95 degrees and then stained. So you see here on the x-axis the intensity of fluorescence and on the y-axis the forward scatter. So this cluster here, this population here are the living cells of that alga. And you see some of the cells in that population are dead or at least old and injured. So around 2% during late exponential phase are not alive or physiological active. Well, and the control, the positive control, we have almost all cells here stained, so that worked quite well. And on this plot here, you see a mixture of both samples. So Tintin did simply just a 1-to-2 mixture, and you see, well, that basically worked out. So we have 48% here dead cells and then 52 living cells. So in principle, this method works for porphyridium. We took the late exponential phase because during that phase, the algae have already a very high amount of extracellular polymers that might hinder cytox to enter or might give an interaction with the stain and the polysaccharides. And that's something we don't want to measure, so no false positive strains. Yeah, so a quick display on pre-study results. Centrifugation before cryopreservation in order to adjust cell density for cryopreservation doesn't have an impact or only little impact on viability. Centrifugation after cryopreservation for thawing and removing the cryo agent has an impact, so it reduces viability, and that's something that is published already, so it's nothing new. And that's something you see here. So we have the control, no cryopreservation, and no centrifugation, and here we have the centrifugation before cryo and here after. An incubation of the CPAs before cryo, so basically that incubation that you have to do before you cool down the cells, that's around 15 minutes, 10 to 15 minutes in the CPA, slightly reduces the viability. And that's something I would expect, actually. So the growth curve you see here is the continuous work of Tintin. We chose different cultivation conditions, rather higher volume. Light path was bigger, so the cell growth is a bit lower than the one from the cells in Scotland. We have 3 sampling points, and that's what we tested. and you see the results on this slide. So I'm going to walk you through that. We have here different cryo agents: diameso, methanol, proline, glycerol, and K1 to K4. These were published to be successful for different green algae. And we have the different sampling points: early phase, exponential phase, and then we had an OD5 sampling point. And then you have the viability. So highest viability after thawing was obtained in samples with sampling point at OD2. In all cryovials, we had an OD1. So the condition afterwards was pretty similar after diluting. But unfortunately, or this is something that other people already observed, sucrose and glycerol containing CPAs promote growth of bacteria after thawing. So even though we have high viabilities here, we see a lot of bacteria that quickly grow and overgrow the algae after the cryopreservation. So that's something we need to take into consideration if we look at these data. Well, so what to do next? We aim at fine-tuning for Porphyridium. We are now using an axenic strain and re-evaluate the cryopreservation and just focus on the most promising ones. And then we also started with different species, like with Rhodella species and other Porphyridium species. Looking at different CPA tolerances, growth, cryopreservation, and evaluate the viability. And something that is still open is the cytox staining validation with fluorescence microscopy, but that's something on hold due to current limitations to visit other labs. And with that, I'd like to thank you for listening, and also thank you for the CCAP team. Joanne and Christine helped a lot, the team in Bremerhaven, and again, Assemble and the organizers for this nice conference. Thank you.
Thank you very much for your talk. So we have a question. Wiebe?
Thank you. That was a nice talk. You mentioned at a certain point that you wanted to try to get axenic cultures of your red microalgae.
Mm-hmm.
Why was that necessary? Because often red algae don't like to grow axenic. They like their bacteria.
Mm-hmm.
Because if you grow them axenic, they can form callus and they don't do anything until you give them their bacteria back, and then they grow on again as if nothing happens.
That's true. Yeah. So, I mean, this is why we actually continued with the, well, let's say, contaminated algae, because they grew better and faster. Yeah, we are aware of that, but we like to see if the cryopreservation, so the growing after thawing, is impacted by this bacterial growth. Because we see high viability in our staining shortly after revival. And if we let them grow, we have too many bacteria in the in the vial or in the culture flask, and the algae grow slower, and it just is reducing the success. I mean, ideally, we find the bacteria that really promote growth and can remove the others that somehow disturb the post-thaw growth. But that's another topic, actually. Yeah, so it's difficult because encapsulation was successful for some strains, but that's not really easy in a commercial aspect, not to do encapsulation method. It takes a while. It has more resources, and an easy and quickly realized procedure is the one that we aim at. But
so, do you actually know what bacteria what are the bacteria? Because
not yet.
Maybe, yeah, because then maybe you could separate the two and inoculate when you need them.
Yeah, so it's a mixture, and well, in every culture, a different mixture. But yeah, I think this is 2 more PhD projects probably to identify everything and figure out who is happy with whom. Red algae, I mean, they like to grow. I mean, most of the algae like to grow with other bacteria in the microbiome, and it promotes growth, and it also promotes the production of EPS, for example. So that's something we are aware of. But we would like to address at least this topic and see if the revival is better without. If not, we learn something.
Very good. Any more questions? There's no. Thank you very much. Oh, one please, Sophie, please.
Hi.
Hello.
My name is Sophie Marot. I work at Villefranche-sur-Mer, and I have a small collection of phytoplankton. I work a little on cryopreservation, and I make some tests with Porphyridium, purpureum, and with DMSO, and 2-step cooling only, and it works well. So maybe with the encapsulation, I discovered this technique a few days ago, in fact. I only use 2-step cooling, but it works well with Porphyridium. Is really resistant to the freezing, even if without GMSO. I have 19% of viability after thawing. So maybe you can try with this kind of method. I don't know for the other genus because I don't test them, and I didn't have them. But maybe you can try like this. It's
so you
really
mean so DMSO is something we have included?
Yeah, without yes. I test 10% DMSO, 15 minutes to incubate before the Mr. Frosty step, and after liquid nitrogen and minus 100 to 50, and it works well. So maybe you can try this. It's easier than the encapsulation.
Yeah, I don't think we're gonna follow up with encapsulation. So currently we're just using 2-step cooling.
And I read in literature, yes, the problem with bacteria after throwing with the glycerol and everything. So it seems complicated. Maybe you can try something easier.
Yeah. I mean, centrifugation seems to be better. So because then you remove part of the bacteria. But on the other hand, you lose a bit of viability. So I guess in the end, it will be a compromise.
Yeah, but if your recovery are great after, maybe if you lose some viability, you can get your better concentration at the end.
Yeah, that's true.
Thank you.
Very good.
It was great. Thanks.
Thank you very much. Well, thank you very much both, and all the questions, and also Inke. And good luck with your freezing.
Yeah, thank you.
Now it's my pleasure to invite Sam Dupont. He is at Göteborg University in Sweden, Kristineberg Marine Station, and of course he's been working on climate change, ocean acidification, and that's what he's going to talk about. Addressing and minimizing the impact of ocean acidification. Please, Sam, go ahead.
Thank you very much. It's a real pleasure to be here and see some old friends in the audience. So thanks, guys, for coming. So today I'm going to try to give like a general talk about the field of ocean acidification, and in particular, I wanted to mention the importance of marine stations in actually doing something like the field of ocean acidification. So not only on how to do the right thing, but also how to do things right. So I will start by a really, really brief overview of what is ocean acidification. So in a nutshell, ocean acidification is a consequence of our carbon dioxide emission. So it's another consequence than climate change. So if you put more carbon dioxide into the atmosphere, the ocean is actually absorbing a big part of it, which is a great service that the ocean is providing to us. But it is not without consequences, because carbon dioxide interacts with water and turns into carbonic acid. And this leads to cascading effect on the carbonate system. So basically carbon dioxide concentration increase, bicarbonate increase, carbonate decrease, the pH decrease, and the saturation state, which determine if the water is corrosive or not, decrease. So you have all these effects all together that is actually potentially stressing for the marine life. And this is something that not only was predicted, it's something that is actually observed all around the world. So that's one of the classic monitoring stations in Hawaii, showing that if you have an increase here in red in the carbon dioxide into the atmosphere, you have the same kind of increase in seawater, and it's linked to a decrease in pH. And the reason why the community started to be really worried about that 20 years ago is because it's happening extremely fast. So you can see here another graph showing the average oceanic pH over the last 20 million years. And you can see that it's relatively stable and high, it's above 8. But what's happening now is that you have this abrupt decrease in pH in the ocean, something about 0.3, 0.4 pH unit expected by the end of this century, which means that the ocean will be 2 times more acidic. So it's really rapid, really fast. And that's why biologists are worried, because species or ecosystem will be pushed outside or their comfort zone. So the global ocean chemistry is changing, that's a fact. The problem is, the next question, of course, is that does it really matter? And unfortunately, the answer is yes. And you can look at the problem from different angles. And one of the angles is to go in the past and check when something like that happened. And the last time that you had a climate change associated with ocean acidification and hypoxia was at the end of the Permian. And at the end of the Permian, 92% of all the marine species went extinct. So of course, I'm not saying that this exactly the same thing is going to happen today, but that shows that we have really good reason to worry. And then the other reason to worry is that now you tend to have thousands and thousands of papers documenting the impact of ocean acidification in field and lab experiments, and more than half of all the publications, in particular on animals, are showing negative effects. So it's about like half of the animals that have been tested in the lab show negative response to ocean acidification. So overall, we have really good reason to worry about it. And moreover, there are places in the world where you can already see the impact of acidification on things that we care about. For example, there's been like a crash in the oyster industry on the west coast of the US about 15 years ago. And that has been attributed to ocean acidification. So it's happening. We know as a fact that acidification is happening, it's a chemical change, and it will and already have biological consequences. And if you don't believe me, you can go into all these reports, including the IPCC report that use this really strong phrasing saying that we are certain as a community that ocean acidification will lead to dramatic consequences for marine ecosystems and things and the services associated to that. And it's also slowly entering into the policy frameworks. And you all know about the SDGs, of course. And it was really exciting when the SDGs were released, because for the first time, the ocean was really visible there in the SDG 14. And if you check what's in the SDG 14, acidification actually is one of the targets. So it's been identified as one of the key issues we have to deal with if we want to ensure that we're going to have a healthy and productive ocean in the future. So the phrasing of the target 14.3 of the SDG is really smart. It basically says we need to minimize and address the impacts of ocean acidification. And that's exactly where we should go as a community now. We should actually work on solutions. So we don't need to document anymore that acidification is an issue. We know that for sure. But what we need to do now is fix the problem. And how can we do that? If you really want to address and minimize ocean acidification, you need to think in 2 different ways. First, you want to be effective. You want to do the right thing. You need to prioritize the science, to do science that actually will matter and contribute to the solution. And the second part is the efficiency. You want to do things right. You want to ensure that if you do an experiment, if you do an observation, you follow best practices in a way that your data will be useful for the community. And in a way, marine stations played a key role in that and will play in a key role in the future. So that's why I wanted to talk about in the second part. And I'm working there in this beautiful marine station of Kristineberg on the west coast of Sweden. And it's the third oldest marine station in the world. And it's been a key partner in Assemble from the beginning. And I moved there in 2005 and to work with that awesome guy, Mike Thorndike, who was also like a key player in the Assemble network. And at the beginning, I was working on other things. I was working on genomics, I was working on stem cells, and it's a really small marine station with like 40, 50 people working there. So it's not that big, but it had a huge impact, not only on me, but also on the ocean acidification community. And one easy way we have to create new knowledge there is through the coffee break called fika in Swedish. So every day, all the visitors, the guests, the local scientists are joining for a small coffee break. And you had no idea how much of a scientific impact this coffee break has. And one example is something that happened at coffee when I talked with Ina and Esther, who were there to work on vision, like they had an antibody to actually target some opsin in echinoderms, and we just talked about it, and I explained that I was doing culture of sea urchin. They were doing some other things on adults, and we joined forces. And just it started with a small discussion at coffee, and at the end it led to a really awesome paper on PNAS on vision in sea urchins. But it also led me to start working on ocean acidification. So you can see this is the number of papers on acidification since like the beginning of the century, and you can see that all happened in the last 10 years. But I was actually introduced to the field by a guest again at coffee break, Lloyd Peck from Bath in Cambridge, who basically told me, oh, you should look that out, that's going to be big in the future. And at the time I was doing larval culture, and I decided, okay, I'm going to give it a go. And I was one of the first, just by luck, just by meeting the right guy. And because of that, I was an expert, and Kristineberg became an important station, an important partner in the field of ocean acidification. So now we are leader in the field, a small marine station like we are, with more than 200 publications, which is a significant proportion of all the published paper. We contributed to best practices. We provide also infrastructure that are used for capacity building. We are involved in different policy documents from the UN, the IOC, the IPCC, and we are a partner in all the major ocean acidification networks. So starting from very little, like a couple of aquarium, a small experiment, we became a major player thanks to this marine station, this position, this attractiveness. So now I think the marine station will also play a key role in the future. And as you know, we entered the ocean decade, so we have really to think about the ocean, the science we need for the ocean we want, and we are really in a hurry. As Greta Thunberg, the Swedish activist, said, the house is on fire, so we don't have the luxury of time. We don't have the luxury to really understand everything. We really need to prioritize the science we need. And if in particular in the field of ocean acidification, we are now at a phase of applied science. So as I said, we don't need more science to document that it's a problem. We need science now to find a solution. So not all science is useful, and we need, all science is interesting, and I'm a big defender of fundamental science, but in the field of acidification, because of the time issue, we need to focus on solutions now. And basically that's the situation. We are the little mouse facing this big issue in front of us, and how we're going to address it is really important. And basically, like the little mouse, we have two options. We can either escape, which sounds really smart, or we can kill the cat. And if you kill the cat, you solve the problem, but it's kind of a difficult task to achieve. And when it comes to acidification, that's the kind of the two same options we have. Mitigation is killing the cat, is getting rid of the problem. And when it comes to ocean acidification, it's basically carbon dioxide emission reduction. So we need to find a way to do that. And we know it's going to be complicated. We know that it's going to require both policy changes and citizen changes and acceptance. And to do that, we need specific science. So that's one of the priority we have. We should provide science that will drive changes in society. And I really like that quote by a colleague from Stanford who said, people have to sacrifice a little bit of their short-term self-interest to help the world be a better place for the long term. And how you get people to do that is the most important research that can be done. And if you want to change people, you don't have to do any kind of research. We have to do research that they will understand and research that will matter to them. And it was in that spirit that we made this experiment a few years ago, where we decided we're not going to do the science we always do. We're going to do science that is society relevant. So we decided to study the impact of ocean acidification on shrimps. And that's the shrimp that is extremely popular in Sweden, the northern shrimp. Everybody loves it. It's in every restaurant. And instead of study the usual parameter, like respiration, growth, whatever, we decided let's go for the taste. So we basically tested if acidification had the potential to change the taste of seafood. And the answer was yes. So actually the shrimp that are raised in conditions simulating acidification don't have such a nice taste compared to the one of today. So we use that as a communication opportunity. And now what we do is we go in shopping mall, we go in school, in public event, and we attract people to us by providing them free food. So we say, come here, we have free shrimps, taste them. And they taste them and realize, oh, these ones are not as good as those ones. Why is that? And then we say it's because of acidification. That's because of our carbon dioxide emission. And we show that by using this kind of approach, people are more willing to actually change their behavior by decreasing meat consumption, being smarter in the way they travel, and so on. So this is just one example of doing science that matters to society and can lead to change. So that's something important to think about. So for mitigation, in a way, if you want to convince policymakers that there is a problem, we have more than enough science. Just check the IPCC report. But if you want to drive emotional changes and something positive in terms of behavioral changing, then you need to think about doing some science that people will understand, connect emotionally and physically understand sometimes. But mitigation is only one of the sides of what we can do for acidification. So reducing carbon dioxide is like killing the cat. The other thing we can do is adaptation. Is basically provide information to industries, to aquaculture, to different stakeholders using the ocean on how they can do things to not be so much impacted by ocean acidification. And a good example is these guys on the picture. They are oyster farmer in Hong Kong, and these guys, all their livelihood depend on the sea and on culturing these oysters. And if they lose their job, they lose everything. So you can actually provide them with information, suggestion about how to change a little bit the way they do things, so they will be able to do that for longer before being impacted by ocean acidification. And there are a bunch of different options available today to make ecosystem more resilient or to adapt industries so they are actually more resilient to acidification. So you can put them in different categories. You have protecting ecosystems. So maybe if an ecosystem is protected, it's going to be more biodiverse and more resilient. So you can do that by creating marine protected areas or reduce environmental stressors. Or you can also repair ecosystems that have been damaged. For example, you can restore degraded ecosystem, like replant seagrasses, for example. And then you can adapt, and adapt is more at the societal level. So you can provide information to aquaculture industry to be more resilient. And just to give you an example of that, this is the story I was mentioning before, where about 15 years ago, oyster farmer in the west coast of the US realized they couldn't produce their little spat, their baby oyster that they needed for their culture. And a scientist called Dick Fitty identified the cause as ocean acidification. And that was a big deal because they were completely out of production for that year. And so research has been done. And first to say, like, mitigation was not really a solution because it's going to take decades before we decrease carbon dioxide emissions. So we needed something faster. And actually, science provided key on how to do things different. So scientists all around the world started to study the impact of acidification on oysters. And they realized that overall, oysters are quite tough. They can cope with the level of acidification that you can expect in the next decades, except during the first 2 days of their development. Because when they move from the trochophore stage to the D-shape, they basically precipitate a lot of calcium carbonate, and there are kinetic limitations. And if you do that under low pH, simulating ocean acidification, they just can't make it and they die. So basically, they took this information and went to the oyster farmer and say, okay, if you pay a lot of attention to these first 24 hours and you do that in controlled seawater condition, then you will be able to produce your larvae, and then you will be able to produce your spat and do your cultures. So what happened is that the oyster farmer used the scientific information to apply that to their job, and actually they are back into business. So that's one example. There are many more about how science can contribute directly to the solution, to adaptation solution, and make things more resilient. So that was for doing the right thing. So, you know, we have a problem, there are solutions available, and you can do your research to actually contribute to these solutions. But the other side is that, no, you know what you want to do, but you want to do it right. And marine stations are amazing for that, because most of the time they already have long-term monitoring, so they are ideal sites to collect information. And also they can be adapted to implement best practices. So in Christineberg, for example, we develop fantastic facilities that allow us to basically invite people or have guests coming and come with their question and do their experiment fairly quickly. So we have all that you need for chemical and biological experiments on ocean acidification, which is not that easy, by the way. You need quite a lot of technique and equipment, because it's always possible to do something. And I'm part of a big capacity building program where we go in Africa and try to set up a lab where people can start doing their research on acidification. And that's an example of what we did in a marine station in Mozambique on the island of Iñaka. So the picture on the top left is basically how the lab was when I arrived. It was a brand new building. The electricity was not connected yet, but within a week, we could actually set up a basic, simple aquarium system to do experiments, and we could collect our first data. So it's possible to do something, even if you're limited in a small marine station. And we have multiple examples now about people starting to do work with just a basic lab. But if you want to do something a little bit more exciting, if you want to tackle questions that are a little bit more complex, then you need better facilities. And in the Ensemble network, we have beautiful places where you can just go and do experiment. This is an example of an experiment we've done a couple of years ago, where we wanted to have this complex system where pH was fluctuating, where you had interaction with different partners like seagrass and calcifying organisms. And that requires quite a lot of equipment. And you can do that in other places like Roscoff, Villefranche, and other marine stations. So the Ensemble network was really also instrumental for this. So if I want to conclude all this, I think ocean acidification is an issue. It's been documented. It's part of the SDG framework now. So we need to tackle that. And now we need to really have a strong focus on solutions. And for that, we need society relevant science if you want really to address the mitigation aspects, so make policy changes and make citizen changes. And you can do science that can contribute to that. Or you can do solutions that will contribute to the adaptation side, so provide information to managers, to industries about how to make the ecosystem or the industries more resilient. And that will allow us to buy some time to take the measures that are needed to reduce carbon dioxide emission. And finally, marine stations are key. And you know, I've spent 16 years in Christineberg now, and I was supposed to be a one-year postdoc, but I stayed because we have there, it's a unique place to work. It provides with endless opportunities. So thank you very much for your attention, and I guess we have some time for questions now.
Thank you, Sam. That was an interesting presentation. Plenty of concepts and policy. You said something at the beginning, which I'm not sure if it was what you intended to say, because I seem to have understood, there's no point in doing basic science. We should go and find solutions, but means implying applied science. But I think probably what you meant is, at least from what followed, is there's no point in describing the problems, but because I think basic science can provide the solutions. You may find mechanisms and solutions based on basic science. I mean, a lot of, the instruments and the mobile phones and stuff came from basic science and so on. So I think probably you meant describing. And I agree completely that we have to be more active now than just, waiting for things to happen.
I think I said that mostly in the context of acidification, but in general, I totally agree that we should fight and keep fighting for fundamental science, because you never know, because it's also creating, the sense of wonder and so on. So I think fundamental science is key. I was more really specific for the ocean acidification field, where no, it's time to do solutions which can involve mechanistic understanding. We can involve, like, really interesting science, physiology, genetic, and so on. And for me, that's still applied in that case, because it's toward the goal.
Problem solving science. We have to basically also spend some time on that. That's curious, because we had this just before lunch. I don't know if you were there, but we had this discussion about industry and so on. And in fact, how infrastructures can benefit industry and how we can get industry to use infrastructures. And in some ways, this is the basics of it, because you have to interest people and you have problems there and you have to solve them. But anyway, so I don't want to monopolize the talk. Carmen Santos wants to ask a question.
Hi, hello, can you hear me?
Absolutely.
Hi, Sam. I was one of the person that you met during one of the FICA summers ago. Thank you for your presentation. It was really great. You're a very nice communicator. I have a question about seagrasses and bivalve refugia against ocean acidification for bivalves. What is your opinion about this as a real solution to create a refugia for these animals?
I think that's an interesting idea, but I'm not sure that everything has been tested yet. So I would be a little bit cautious about, like, if it's really the way to go or not. Actually, we tested that with seagrass. The thing is that if you put seagrass and kelp in high density, what's going to happen is you're going to have use of carbon dioxide through photosynthesis, right? I think that's a little bit the idea, and you capture part of the carbon dioxide. And that works, of course. The problem is that it's creating a lot of fluctuations too. So during the day, you're going to have high pH. During the night, you're going to have low pH. And it hasn't been addressed properly, because if you create more fluctuation than there is today, the problem is that organisms respond to extreme most of the time. So if you create lower pH than actually what has been there before, that might actually have detrimental effects for some of the calcifiers. And I'm not saying that this might always be the case, but that should be tested. And actually, with the picture I showed in my presentation, it's exactly what we tested. So we created natural variability by high density of seagrass, and we tried to check what was the impact on the calcifier there. And the interesting part was that the calcification followed exactly the same pattern of the fluctuation created by the seagrass. And the consequence for fitness, we are not done analyzing the data, but I can probably tell you more in a couple of months to see if it was good or bad for the creature. But I think it's a really good potential idea, but we should test it properly before going full scale, at least if your idea is to protect bivalves. If it's for other things, I think it's great. I think in general, it creates biodiversity and so on, so that capture carbon, this is fantastic. But for calcifiers, there might be a dark side.
So I have a question related to that. Do you have any information about what could be the impact of bivalve aquaculture? Basically, take away the shells, and eventually you can use the shells into some way that it doesn't dissolve, So I think you are removing, but I'm not sure how big this removal is in terms of what impact it could have.
Yeah, I think some people are exploring that, actually, like using all the waste from the leftovers from mussels, from oysters, and so on, and try to put that. And it seems it can have a positive effect. From the experiments I've seen, it can have a small positive effect locally if you do it right. But it's challenging practically to actually make that happen. But that's an idea that is explored for sure. But there's also a dark side from extensive oyster and mussels aquaculture, because these guys are calcifier, and there is this misunderstanding that actually they are capturing carbon through calcification, but actually they produce carbon. So if you have heavy calcifiers like mussels, oysters, or even corals, overall the budget is positive. So they produce carbon through respiration and the calcification process in itself. So that's also another thing. I think you're muted.
Sorry, I was saying, so it seems as if it may not be a solution. Although it may not be as bad as other things, basically. But
that's always trade-offs anyway at the end of the day, and we also have to consider the other aspects. But yeah, I think there is no silver bullet when it comes to addressing that. I guess we're just buying time.
Yeah.
And there are some options for that.
No, for sure, we have to produce less CO2 as well.
Mm-hmm.
Well, I don't see any questions. Anyone else wants to ask a question? No? Well,
that was great. Thank you so much for the opportunity.
Thank you, Sam. It was nice to see you, and thank you very much for the talk. And I'm going to just, okay, I just thank you from Joanna. Yeah, thank you very much, and thank you for the audience to be here and listening. To tomorrow morning, we will have more talks. We'll start with Colin Brownlee, and then carry on with some presentations by marine stations, and followed by some other talks by Assemble Plus users. So it will be a long day tomorrow, and like today's a bit shorter. So thank you very much, everyone, and hope to see you again tomorrow. Thank you, Sam. Bye.
The marine environment is a rich and largely unexplored reservoir of biodiversity with vast potential for food, health and biotechnology. The European Marine Biological Resource Centre, EMBRC, is a research infrastructure which aims to unlock the knowledge and innovation potential of our oceans. It enables researchers and companies to access marine organisms, expertise and experimental facilities to study them. Headquartered in Paris, EMBRC brings together 45 sites in 9 member countries.
We provide access to specialist facilities and services that enable researchers from academia and industry to study marine life and develop innovative solutions to address societal challenges like climate change and health and food sustainability. We support both fundamental and applied research, particularly for areas like biodiscovery, biotechnology, aquaculture, biodiversity, and climate change research. EMBRC-supported research has already led to novel, high-impact research in human health, product and medicine development, and aquaculture, and it's helping us to fully grasp the crucial role of ocean life.
EMBRC has benefited hundreds of researchers across Europe and beyond, delivering robust and efficient services and expertise to help users obtain the best possible results.
So EMBRC is continuing to develop its services. We're working to start recording biodiversity at many of our sites using molecular techniques to put in place so-called genomics observatories. This will allow us to have a much better understanding of how our oceans function and their current health. In addition, we're increasing our bioprospecting capabilities to better support the development of new products and solutions from the sea.
EMBRC is a single access point to remote and on-site services in Europe, supporting marine research and innovation across borders.